So welcome today to the second webinar in our Translational Digital Biomarkers in Drug Discovery and Development webinar series. This is a 4-part webinar series, and today we have 3 excellent presenters who are going to talk a little bit about the realities of onboarding these new technologies. In case you missed it, we did do kind of an introduction overview of translational digital biomarkers in our first webinar, and that recording can be found on our website. After this, we're going to have a webinar talking specifically about study design, so stay tuned for information on that. So just so that everybody knows, you will want to submit your questions using the chat. So you should have a little thing that looks like this. You should be able to click this little chat button and then type your message here. We'll answer all questions at the end, and then all attendees should be muted and have your cameras off, which is an automatic setting. So the North American 3Rs Collaborative, who are we? How are we organizing this webinar? Well, we're a nonprofit whose mission is to advance science, innovation, and research animal welfare. And we do this through facilitating collaborative 3Rs efforts focused on refinement, reduction, and replacement. We really try to partner with individuals across the field. This is a picture of our leadership team and board of directors, as well as their institutional logos. And I hope that you can appreciate that we do span academia, industry, government, other nonprofits, biotech, CROs, suppliers, and more. And we do this in our initiatives as well. Our strategy is really to identify initiatives that we see that have strong evidence, will make a big impact, and ultimately have that real-world practicality piece. We currently have 6 key 3Rs initiatives focused on rodent health monitoring, microphysiological systems, translational digital biomarkers, refinement, creation of a 3R certification course, and compassion fatigue. And if you want to learn any more about those initiatives, you can go on our website. We also have a recent webinar on compassion fatigue that kind of gives an update, and I will be conducting a fall webinar as well. But of course, today we're talking about translational digital biomarkers. This initiative collaborates to increase adoption and regulatory acceptance of translational digital biomarkers that refine and reduce the use of animals in research. We actually have 16 different technology providers and end users and other agencies that are members. You can see all of their logos here. And generally what we try to do is we try to collaborate to establish improved understanding of the value of these technologies, to facilitate optimal implementation, to engage end users and others interested in learning more about how these technologies can be used to promote the 3Rs. We have some resources. So we did publish a manuscript early this spring that has a checklist for evaluating technologies, really used for figures. Again, you can find this on our website. We're working on creating more educational resource pages and regular posts. We've been presenting at these webinars, and we will also be at ALAS. And finally, we're going to be creating a technology hub of available systems that's easy for end users to engage with. So we've got 3 wonderful speakers today. Matt Ruiter is going to start off talking about vetting translational digital biomarkers. Then Ellie is going to talk about operations and infrastructure realities. And finally, Paul's going to round us off with realities of onboarding. And with that, I'm going to turn it over to Paul.
Hey, Megan. I think Matt is starting to.
Oh, wait, no, I'm going to turn it over to Matt. My apologies.
Thank you.
Thank you, Paul.
Yeah,
Matt.
Turn my camera on. 1 second. Can you hear me, Megan?
Yep, we can hear you.
Excellent.
We can
Thank
see
you
your
so much.
screen.
Excellent. So my name is Matt Ruiter from UID. I am one of the tech providers of the association. So I'm going to talk a little bit about vetting the digital biomarker technologies from kind of our side going forward here. And when looking at technologies, I kind of want to focus today on three factors: scientific, strategic, and IT. I'll get into each one of these a little bit of detail in the next few slides here. So scientifically, you first want to determine the use case and what does success criteria mean to you? Is your justification, is it scientific? Are we looking at a particular biomarker endpoint, maybe as an activity, or it's a grooming pattern, or is it husbandry? We're looking at how often do we want to change the cages? Can the system tell me when a cage is dirty, or how much food is in the system? Or maybe it's both. We combine the scientific and husbandry to look at that. We want to prioritize the endpoints. The success criteria, what does that mean to you? It's very, very important, so that when you're looking at technologies, you want to bring them on board and understand what they can do. What does that success mean to you? Again, is it scientific, is it husbandry, or both? Bringing those together here. But please know that one system can't do it all. We wish we could as tech providers, we wish we could, but again, each of the systems out there that are on the hub all do it very, very well. But again, one system won't give you everything you're looking for. So when you look for your endpoints, it's not what you want, it's what you need, right? That's make that very, very important here. Strategically, we want to align these programs, the digital biomarker, with others in your facility, right? So the first part is, who is the technology owner? Is it going to be you as a PI, as a researcher in a group, or maybe it's a team, maybe it's a smart cage team within your facility or university? Then how can this be used throughout the facility? So other researchers on board to gain widespread adoption, right? So when you look at bringing the technology on, is just for you, or strategically look how we can improve the broad-based use throughout your facility. And this one here is key. It's the key for us here, IT. So IT is something you want to engage early, and I can't stress this enough, is communication with IT is very, very key. And again, I'll explain more in just a slight bit here. So first I'll talk about is the company-wide adoption and how we can improve internal approvals. So scientifically, does the digital biomarker or data that you're looking for, does that support your experimental goals? Does the technology match what you want in the end here? So when we do that, we're looking at how do we design a study. And Megan mentioned earlier that there will be a whole series on this, but just as a broad 10,000 foot view is let's look at what the technology can offer and how do we define an experiment to bring this technology on so we can vet it to make sure it's the right technology for you and for your company here. We also make sure we're comparing apples to apples. And what I mean by that is a good example is locomotor activity here. So if we're looking at locomotor activity here, we have beam brakes, we have video, we have RFID, disruptive field. These are all technologies within this group here. And they all do it well, but they're all different, right? So when we're looking at single versus grouped house animals, the activity level is going to be different when you compare those two. So when we're looking at that technology, make sure that we're keeping the data that we're looking for consistent within the group, the animal, and or the cage or the rack systems. Again, so comparing the technologies to themselves, not to other ones in the field, right? That's very, very important to look at. Strategically, what I mean by that now is that when we're looking at either scientific or husbandry, how can the data be collected and useful to other researchers in your facility? I look at this as kind of the water cooler talk, right? We don't have water coolers anymore. They're kind of gone with COVID here, but generate buzz, right? Generate buzz so that you get others interested in this technology. You may want it for a very select reason for your experimental design, but if we can get widespread adoption, that's going to help increase utilization of the technology to help with the ROI, right? Everybody's concerned about how do I, return on investment of this technology, and that means a lot to a lot of different people. So a scientist in the group here, you're looking at data. Data is very, very important to you. So that's your ROI, right? Others may be a vivarium manager when they say, I need to basically reduce my number of cage changes. So that's their ROI. So whoever's writing the check at the end of the day or the PO to the vendors, make sure that you understand what the ROI is and how you can utilize that throughout the facility so it makes it easier for the person ultimately in charge to sign off on that final system here. And then have realistic expectations of how long it's going to take, right? So once you know what you want and you have other people's on board, how long is this process going to take from talking to a vendor, looking at trialing the system, looking at the system, see what's best for you. Is it the right technology? Maybe you're trying 2 or 3 different technologies. No one understands this is a long process. This is not something we just put in overnight. It takes a lot of work on your part from the internal scientists as well, and also the vendors to onboard this technology. And sometimes we're talking anywhere from 3 months to almost a year to bring it in. So again, understand the timeline so that everybody has expectations. They're realistic going forward throughout your facility. IT, this is always kind of a love-hate relationship. So if there's any IT people on the call, thank you for being here. IT's job is not to say no. All right, it's usually what they say when we say, here's my PC, I want to plug it into the wall, and I want to access their data remotely. They're going to say no, obviously, right? Because their job is to secure the technology insight. So understand the role of IT in your company. What do they do? They're not just there to fix monitors that don't turn on. They're there to make sure that the data is safe. So engage them early. A lot of times we see that IT is the last people to bring on board, and they can accelerate or they can stall this process very, very quickly here. But communicate with them that how significant their role is in this vetting process, onboarding technology. IT is very, very important to it. We have to look at how you access the data. Is it going to be static? That's maybe on a unit in a room. That's going to be very little IT involved, but again, very important. Or do you want to access it remotely from your home? Where we're going to store the data? Is it on maybe a vendor's PC, or is it on their data center? And where the backup's done? All this has to be done before you get the unit in the system in your facility here. Their job is network security, cybersecurity, so they're very concerned about data going through their system here. And then ultimately, how does that system communicate? Is it Wi-Fi versus Ethernet? And then look at cost of implementation of the system. Data storage can be expensive. We can buy a terabyte drive from Amazon for a couple $100, but that same drive to put in the data center is completely different and a lot more expensive. Cloud storage, we know, Azure, AWS, gets very, very expensive. And the technology that we have today, it can generate a lot of data, right? You're talking about, terabytes of data here. So what does that cost? Add that to the cost of the equipment and bring it on here. What is the cost for IT support? Where is it going to who's going to be in charge of that as it goes forward? So once you have the system in place, what does that support look like, right? How much is it going to cost here? And what additional hardware do you need? Do you need a new server? Is it going to be on a virtual machine, which is doesn't cost anything, but it takes resources, IT time. So again, engaging IT early will definitely help with that process. I want to give a quick little case study here. This is we did an install at a government facility where we provided a PC with a custom Windows image on the software. So that Windows image was designed to work with our system, the communication protocols and the security over here. But however, as a government facility, you can imagine they have their own requirements to bring in there. So it required us to put their image on one of our PCs. So to basically they wanted the data on their data center here. So you can imagine when you do that, the Windows images kind of don't talk right here. So we had a lot of issues with the communication being blocked. It wasn't all the data, but it was some packets being missed, and we're trying to figure out why. So this is why I said engage IT earlier and let the vendor and IT, as well as you personally, work together to find solutions. So ultimately, the solution was that the software had to be changed a little bit to work with it. But it was very important, but to have the collaboration and flexibility for all the parties, right? So sometimes IT is kind of third party, sometimes they're managed offsite with the vendor, and you as the researcher looking for the data, you want the data, we get that, but we want to make sure that we support the IT side as well to make sure that the data is being transmitted how it's supposed to and everybody's happy. So again, that collaboration with IT early on definitely helps with this process. What is your definition of success? This is very, very important. And this is something that when you're looking at technology, try to define that as best as possible. I know it's going to be hard, and it is very hard to do, but have a defined series of questions that, when you're looking at technology, what can that technology do? What can it give you? And design your study around that. But also know that since these aren't new technologies, but they're being now used in new ways, right? It's getting a lot of popularity, and people are using them more and more, and we're getting more research done that we never knew we could in the past. So understand that the data that you generate may challenge or maybe change your hypothesis. It may be something we want. A plus B may not equal C, it may equal D, but D is very, very important to you. So when those goalposts move, as you're looking at the technology and you see what it can deliver to you here, be prepared to say, hey, this is great. I like it here, but also now we need to switch and do a new study. We all go back to what is your definition of success? Because obviously, again, I said earlier, one technology can't do it all, but your success may be rooted in some part of that. So again, look at the technology, what can it afford, and understand what does it mean for you to be a successful vetting process. Some quick other factors to consider before I pass it off to Ellie. is going to be software usability. This is often overlooked a lot of times. Again, it's a very high level talk I'm providing now, but is the software intuitive? Is it easy to use? Where's the training come from? How easy is it to be trained on? These are all things that can be overlooked, and you don't often know this until it's been too late, right? So talking with the vendors beforehand helps you through this process here. Who's going to do the data analysis? These are large data sets, right? These can be very, very large data sets and up to interpretation. Do you have data scientists on board? Do you have statisticians? Do you have a team that can help you with that? Because the vendor algorithms, while they're good and also very, very well tuned for what they do, they may not be exactly what you're looking for. So how do you then take the raw data set and use your internal teams to help you understand the data? So software usability is a very, very key important aspect to look at when you're vetting these technologies. How scalable is the system and how adaptable is the system being evaluated? If we look back at 2019, who would have thought we'd be here now in 2022 with COVID? What will we be doing in 3 years? Your guess is as good as mine, right? We have no idea what's going to be happening here. So how can the system that you're looking at be adaptable, and can you add technology to it? Can it increase with the times? If you do, maybe you're doing one set of research now, but in 3 years you're doing something completely different. Can this technology grow with you? Can it grow with your research demands? It's very, very important to look at the future aspects of it and how long it can be used going forward. And lastly, what does the post-installation support look like? Staff changes, right? Especially universities, you have new staff coming in all the time. So does the vendor, do you as a group support new training of staff here? Integration, new technology, like I just talked about here, software updates, how are those given? Because those kind of go back to where IT gets involved. How are software updates given? That's very, very important here. And ultimately provide feedback. So when you're vetting these technologies here, I look at it as one of the best Clint Eastwood movies, The Good, the Bad, the Ugly. We want to know it all. And the more feedback that we get, that we work with you and through IT here, we to enhance this technology so that it's ultimately the best thing that we can provide as vendors, as technology providers, to the researchers to make the process as best we can. Thank you very much, and I think I'm going to pass it off to Ellie now.
Thanks, Matt. If you guys could just confirm that you can see.
Yep, we can see your
my slide.
Yeah, looks great.
Okay. And the correct slides and not the speaker notes.
Yep, you're
okay.
all good.
Thank you. Hi, I'm Ellie Karlsson. I'm the director of the Lab Animal Resources Group here at Calico Life Sciences. And while I'm a lab animal veterinarian by training, I've spent many years in my career onboarding various technologies and integrating them into the vivarium infrastructure and the animal program. And so today I'm speaking specifically to operations and infrastructure, but you'll see throughout the talk that there's a little bit of overlap between what Matt, myself, and Paul will be presenting. So it's important to know that smart caging will impact your vivarium space in terms of the animal holding room. And it really depends on the technology you choose and what the footprint of that rack looks like. So here I have a couple of examples of like a UID Innovive rack versus a Sable Prometheon metabolic cage system. While both are scalable rack systems, there's varying degrees in the capacity of cages that each of them carries. There are also tabletop designs versus rack designs. And so you're going to want to think about how these rack systems will fit into your vivarium. There's also varying degrees of housing for the mice in the cages. And so, if you're going to be single housing mice versus group housing, that really impacts how many racks you'll need to run a specific study. Some technologies also have accessory or ancillary equipment. Our Sable metabolic cages, they need gas tanks to help regulate the gases. Might come with computer towers or separate blowers. So for those of you who are in the space planning, really need to dig deep, and you might see a brochure of a rack, but you want to make sure that that's really what it comes with, so you can see how it fits into your space. And so all of these things will impact your scalability. Another thing to consider for animal holding room is, do you dedicate one room to digital technologies, or do you integrate these smart cages into your standard housing? And that really depends on the scale of your study, also the type of research that you're doing. So for those who might be interested in circadian rhythm, they might not want a room that's entered very frequently by others. They might want something dedicated. And so this is an important discussion for you to have with your scientific team as you plan for these technologies. One unexpected impact that we felt was how smart caging impacted our support space. One case example is how much space do we need to store cage setups or these accessories within the vivarium? Because these are not parts that you could just jumble in a bin, you generally have to build up the cages and have them ready for experiments. We found that we were using multiple metro racks to set up for experiments and sort of be on the ready when our research team needed to do recordings. Another thing that we didn't consider is how many additional or backup parts are needed, especially if you're running high throughput studies. So for our institution, we have 64 Sable cages, and on occasion, some of the sensors might not work, or you might need to run back-to-back studies. And so when we initially had just 64 cages, we ended up scaling to 1.5x. So that way we always had some overlap in the event that we needed to run studies in a short time span. And then also, how do you prepare for malfunctioning parts? Some of these sensors cannot be interchanged between modules, and so you're really going to want to try to understand how interchangeable parts, cages, and systems are when you're doing your purchase of the system. All right, smart caging will also impact your vivarium sanitation processes, and that's because not all smart cage accessories can be run in the cage wash or autoclave. Some of these things could either be highly sensitive and need to be hand sanitized, and you really have to consider what alternative sanitation intervals you might use for single housed mice. So this here is a picture of our Sable parts. And we actually run mostly an Innovive institution with disposable caging, and we don't have access to a tunnel wash, so we had to get equipment to appropriately sanitize these, and that was a challenge for us. And so in terms of exploring your alternative sanitation processes, in a facility with no cage wash, do you have places to hand wash? Do you have spaces to dry or store individual components as well? And then lastly, we really had to work hard to verify our sanitation processes because we weren't getting up to 180 Fahrenheit, which is the guide requirement. So we went through many testing cycles to see which disinfectant, contact time, and testing methodology would verify sanitation of these specialized cage parts. Now, you might have to look at your upgrades of the vivarium before you bring the technology over. And so some of the things to think about in terms of your facility upgrades, rooms may need either Ethernet or Wi-Fi. I think that's very difficult in some of these older facilities where it's very hard to get signal depending on where your vivarium is located. For things like video or high volume data sets, having data drops installed in the rooms is something that took time and resources to do. You'll also want to consider additional power outlets, as well as how soon does that emergency power kick in so you don't have loss of data. Another consideration for our team, as well as the researchers, is how are they going to interact with the new platform? So Matt touched on this a little bit with the software. However, when we're working in a vivarium and people tend to be very mobile, are they going to be walking around with a laptop? Is the user interface only accessible through a desktop or laptop? Or is there an option for tablet or even mobile phone web interface? And so those are the kinds of things that you'll want to think about. And then depending on the health status of your facility, do you want your staff walking around from room to room with those devices? Okay. Translational digital biomarker technologies require support from lots of different teams. And when we're onboarding, we think about things like who will operate and maintain these systems. Oftentimes the installation is pretty straightforward because your IT team supports you there. There's some infrastructure that they need to, hook up the equipment. However, as you start to use the equipment, who's responsible for preventative maintenance? During the study, your scientific team may be more or less responsible for setting up the experiment on the user interface and managing the technology on that back end. However, consider if that technology requires any type of calibration before the study starts. And then very important, especially if, I think just all the time, monitoring the data output and quality. Sometimes, things can go wrong if a running wheel isn't properly greased and it's not rotating at the normal speed. There may be aberrations in the data. And so it's important to know who is looking out for these aberrations so that way you run a successful study, and who can respond to alerts, not only looking at the cage and the animals, as well as who's going to pinpoint what the problem is and troubleshoot that. Now, during the study, the animal care team is really essential in supporting these studies. And that's because they're still, for the most part, going to be looking at animal health as a part of their daily health checks. However, there are some smart technologies that send out health alerts and say, hey, activity is down, or there might be a sick animal. And so the animal care team or the veterinary team really needs to be in tune with how these alerts work and set up a system for how to respond. And Paul will be talking about that later. On our team, our animal care team is heavily involved with the setup of these instrumented cages, as well as with the interaction of the software. And this is where involving them during the onboarding and getting their input, as well as developing a robust training program, was very, very important. We found that our animal care team was very excited to interact with this technology and be on the leading edge of refinement with animal studies, and so they were very eager. Now, that's not to say it was easy, because many of them may have only had experience either sending emails or using barcode scanners to send communications, and so navigating a web interface and entering in data was new to them, but it was definitely accomplished with a lot of training. And then lastly, this is where our animal care team is mission critical, and that's in ensuring data integrity. As Matt mentioned, there's a vast amount of data that's collected from these studies, especially if it's 24/7 and you're doing a longitudinal study. And you want to make sure the data is reflecting the correct animal, the correct study, the correct study group. And so we spend a lot of time verifying in our facility the identity of the mouse, the identity of the cage, and ensuring that the study, as it's reflected in the user interface, is what's actually in front of us in the mouse house. One case example in terms of requiring multiple teams for digital technologies is how do you integrate this new technology into your vivarium workflow? And in lots of discussions, there's this general fear that once we adopt technology, we hear things like automation and we're not using people anymore, that there may be less opportunities for our technicians and our researchers. However, I've also found that when you onboard technology, you can also identify lots of growth opportunities within your institution. And so our animal care team and our in vivo researchers directly contribute to the onboarding of the tech, because if it's not going to be implemented in a way that's user-friendly for them or for us, it's not going to be something that we can scale. And we are all invested in the success of technology, that addresses the 3 Rs and gives us good data. So we're all aligned in wanting to onboard this properly. There's, again, opportunities for ongoing monitoring and reporting of systems. So, animal technicians are very adept to doing health checks and looking at food and water and facilities and temperature. Well, it's no different for technology. Is the user interface working? If you log into the app, does everything look correct in terms of the streaming of data? Does everything look like it's hooked up correctly? So there are people within your team that can become subject matter experts in assessing these scalable technologies. And then lastly, implementing lots of process improvements throughout the onboarding and use that increase the scale and efficiency of digital experiments. In our institution, we started kind of small, and then once people started to understand the value through pilot studies and other presentations, we started to use it more and more. And so with every study and iteration, our teams were critical in terms of putting together, a tech request system to set up the cages, a schedule to take down the cages, and then communication to let folks know when studies were happening. Now, this is a recent paper that was put out in Nature back in April. So it's about using multi-pose animal estimation, and they're identifying animal motion in a group house setting using individual cages. So this is very early stages, and using a lab-based software called Deep Lab Cut. This is not by any means an example of what folks are able to do now, but for context, I want you to know that I live in the San Francisco Bay Area. I grew up in the Silicon Valley, and where I live in San Francisco, I'm surrounded by driverless cars, and not just one driverless car. I see 2 or 3 makes and models pretty much every time I cross the street. And I always get the sense that I'm living in the future a little bit. And so when I look at this study of what we're able to accomplish in this recent study with tracking animals and estimating poses, I think opportunity. There's people who are paid to drive these driverless cars to train the machine learning models. Well, I think in this case, who better than our animal care staff and our researchers who understand animals, animal movement, and animal health best to help propel this technology into the future. And so my prediction is, at least within academia and within some companies, there will be future opportunities on doing annotations on animal videos and photos. Certainly within our institution, we engage our technicians and researchers on pilot studies where we are comparing traditional methods versus digital methods and outputs. And so I just wanted to put that out there, that this is an opportunity for many teams to get involved in this technology. And with that, I will pass it back to Matt.
We'll turn it over to Paul now.
Oh, sorry.
Yeah, no worries.
Yeah.
Apparently, we just want to call you Matt today, Paul. So
It's all good.
for whatever reason.
I don't mind being Matt, that's for sure. Right,
Yep.
okay, everybody can see. Okay, let me just duplicate this slideshow.
Yep.
Great.
You're in presentation mode. Go ahead.
Set and running. Great. So my name is Paul Makidon, and I'm the Director of Comparative Medicine in the West Coast for AbbVie. And in addition to being a veterinarian, I've been heading up the science and trying to onboard digital technologies into the vivarium of the future, I guess, for AbbVie. In addition to my veterinary training, I'm also an electrical engineer and a biomedical engineer, so I naturally kind of gravitate towards kind of this geeky stuff. So appreciate your time and your attention to this talk. This phase of the talk is going to focus on onboarding. How do we actually onboard the technology once it's vetted? As Matt went through, once you had your considerations that Ellie discussed, now it's kind of the rubber meets the road. How do you actually onboard it into your system? And the reality, and as Matt and both Matt and Ellie touched on, this process to onboard this technology into a pharma company, for example, can take a significant amount of time, especially if you're looking at using the data for regulatory submissions. So there's quite a few validation verification steps that are absolutely critical. And estimates are anywhere from several months to over a year, and it depends on the use case scenario. And if you're adopting one technology, if you're adopting multiple technologies, but there's a couple key critical points here. And one is you need to, in the front end, to be able to establish your criteria for how each of the steps of the validation is going to occur, all the way from vendor onboarding through actually conducting the studies, the pilot studies, and then validating clinically. And the second key point here is that you have a great understanding of the time frame of this, and you share this with the stakeholders who are enrolled in the process, because this can take quite a bit of time to happen, and you want to make sure that your stakeholders are looped in and have knowledge of the time frame, and that you can align the support mechanisms that are necessary to move each of the phases forward. The process of verification and validation is multifactorial, and you can kind of split it up in phases. So the technology verification itself, whether we're talking that it's done on the ground at the tech provider or once you have it on the ground in your facility, this phase is often completed by the manufacturer or the tech provider and or in concert with the end user. And this is the phase where we're actually making sure that the device itself can turn on, it can communicate, it can measure, and it can transmit data correctly. And also moving into the precision of the equipment. Is it reliable? Is it reproducible? Is it functioning as it should? This is before any animals touch the equipment. And this can take quite a bit of time when you're talking about a system that is shipped and installed in situ into your facility. There's a lot of different factors that may make the setup of this an interesting process. In the analytical validation phase, this is where the end user is typically working with the tech provider. And this is where the actual algorithms are being generated, and you're looking to see that you can convert signals from the tech platform into measurable events or output metrics. This also takes quite a bit of detail. It is a effort that involves collaboration of multiple different teams: IT, animal care staff, the research staff, the tech provider, and can take a considerable amount of effort to get the analytical validation done, especially with the analytical teams that are evaluating. Now, there's options as far as analytical development, whether you're using kind of out of the box setups as provided by the tech provider, or whether you're using internal teams or a combination of the two to get that all set up. Finally, the clinical validation phase can often be the lengthiest process. This is where you're really looking at the robustness, the reliability, reproducibility, and usability of the tech. You want to make sure that the digital outputs that you're measuring are predictable for what's going to happen clinically, or at least give you functional, biologic, physiologic, or functional states in the preclinical phase that will dictate or give you early go, no-go decisions, or help you understand how things may unfold in clinical applications. This phase, it's extremely important that you're kind of running a head-to-head comparison or a gold standard study, where you're looking at your traditional methods of measuring outputs that impacted pipeline decisions. So this can take quite a while to go through. It is absolutely critical that on the ground animal pilot testing within your facility is conducted, this you need to carve out a bit of time for and a bit of budget for. It's not as easy as putting the setup in your new vivarium facility and kind of turning the switch on and starting to run studies. You really need to have an understanding how each of these different platforms work, and it's dependent on the use case scenario of what technology you're using, what you're trying to measure, so on and so forth. So acclimation is an extraordinarily important part of that process. You're all aware, traditionally, you want animals to acclimate to reduce stress and to get them familiar with their surroundings. Well, kind of the same thing here. This is novel equipment, and it may present novelties in, from traditional housing standards. And that can represent change for not only the animals, but also the animal care staff, and can impact husbandry practices. So when you're acclimating animals, you want to make sure that you give them enough time, and you give the husbandry staff enough time, and the research staff enough time to really develop a familiarity with, with the new setup, and that'll help you establish baselines that'll be meaningful for you. The baseline establishment is something that also is critically important. In our experience at AbbVie especially, we often establish baselines over several days, if not weeks, to understand the total arch of the variability that can happen. And I'll walk you through kind of a case scenario of that. There are opportunities for folks to come into the room, at different times, say they forgot a piece of equipment or they wanted to check on an animal. Any interaction that, that staff that's entering the room, can create these data spikes, which can cause variability. And these are important to understand when you're developing your baseline. It's important that, that you allow enough time to kind of see all of those scenarios that can develop and understand them. And that you're giving enough time to evaluate the equipment function and reliability. Finally, establishing the controls. And this also feeds in from the baseline. There's several critical factors to consider for controls. Are you using a group control? Is the cage itself a control? Or are individual animals a control? And it really, again, depends on the case use scenario. Some studies and some digital outputs that you're measuring, you take activity, for example, you can have a group of 5 mice, and there can be animals within that group that can be much more active than other groups, or than other animals in the group. So really taking the time to identify, based on your baseline establishment and your acclimation period, what the most appropriate use of controls are that will enable you to not lose data and to develop these sensitive outputs, that will help guide your pipeline decisions. So establishing great controls will be key for developing your sensitivity and specificity, and to help you power these studies. In most cases, if not all cases, the animal care staff is going to be absolutely essential in this process. And Ellie mentioned this. You're going to want to enroll this team very early. Even through the technology onboarding and validation, Ellie mentioned that this team will require a lot of training. They're often very excited and engaged to do this. This is something that allows them to, interact directly with the animals, but also with the technology. And so enroll this team in the study design, study setup, and execution. Have them right there in the meetings with the research teams in the therapeutic areas that are doing this, because they're going to be the folks that are often really operating the system, doing all the troubleshooting, doing the study documentation. I mentioned data spikes and people who are entering rooms. This is where metadata becomes extraordinarily important. So establishing all that documentation of anything that is happening that could impact the system, being able to record that and have that metadata available. So if you're seeing aberrations in data, you can go back and you can reference the metadata, and the animal care staff team is one of the most essential components of that. As Ellie mentioned, the bringing this technology into your facility will certainly impact operations. And one of the considerations here is cage changing, routine husbandry practices. Is this going to alter? Well, it depends on the technology that you're using. Certainly, the idea of entering a cage, changing it out will impact your data. So again, metadata recording is very important. But establishing the timing of this, and some of these technologies, some of these smart caging systems are capable of monitoring things like water levels, food levels, waste levels, and you can use these systems to be able to help understand the best timing, to change cages. When you change a cage, is it important to recalibrate the system? For example, if you have a computer-based, computer vision-based system, do you have to recalibrate the cage in order to continue to measure after you change, the bedding and the settings in the cage? That is all dependent on the technology that you're using. Some critical factors to consider: the choice of nesting and bedding. I'll bring up the computer vision based scenario again. It's very important that you're able to get high definition resolution with these type of systems, and you need a lot of contrast. So when you're choosing your bedding, the technology may be impacted by that. What's going to provide the best contrast? What's what type of nesting is going to obscure the kind of the micro movements that you're interested in measuring? All of these factors are important, and you need to work closely with your tech provider and in the onboarding phase and figure out what's the optimal system for you to be able to have the most sensitive outputs. Housing density. Some of these systems are dependent on the density of the animals in the cage. Some of them, the less animals, the more sensitive it is. Some of them are not that way, but you need to determine that. You need to work with your vendor there. You need to do the appropriate pilot testing to figure that out. In some instances, you may need to single house. Light cycle impacts. Again, going back to the computer vision, illumination may be necessary to be able to generate the photographic or video images. So will there be impacts on light cycle on these animals? Again, it depends on the case use scenario and the type of equipment you're using, but something that you definitely need to consider. Things like heat load. These equipment can generate heat and vibration and things that will impact husbandry operations. So these are also things that your operations teams should think about. And Ellie mentioned the impact on storage. There are several issues that could relate to regulatory impact when you're thinking about bringing this technology on board. I mentioned one of them. So single housing may be necessary with some systems. So these are things that your IACUC bodies will have to consider and grant permission to do. For example, some systems, because of the need to measure video output, you may not have the luxury of using certain types of enrichment. So exceptions to enrichment policies will need to be considered by regulatory bodies. The IACUC will need to consider things like potential stressors, circadian rhythm disruptions in the cases that may be noise, vibration, if there is any, and whether you need to transition these animals between instrumented and non-instrumented cages, the stress associated with that. So these are additional factors that IACUCs will need to think about. Here in our facilities, cameras, recording devices are not allowed. So now we're talking about putting live streaming, video cameras that are 24 hours a day in our system. So working with security teams, working with the IACUCs and coming to an understanding how we can secure this data and information so it's not falling into the wrong hands. Ellie mentioned this as well. Smart caging will impact veterinary care. There's some exciting opportunities here. Can this technology help us develop digital surrogate endpoints that we can use to even offset animal care staff evaluation? Will these be more sensitive than peering through the cage twice a day? The jury's still out on that, but there's some exciting possibilities there. And speaking of that, veterinarians will now potentially have access 24/7 to live streaming data. 3 o'clock in the morning, I could get a notification from one of our smart cages that an animal is reaching an endpoint. So what does that do to me? What is that, ethical obligation there? These are things that, organizations will need to really have great discussions about and understand, how that organization is going to handle that information and what's the, most appropriate approach to animal welfare given, the access to the data. The great news is that, this technology has lots of utility for 3Rs potentially. We can use it to reduce handling in some cases. We can perhaps better manage the assessment of pain and stress. Can we come to more earlier endpoint analysis? Can we be able to measure subclinical disease more sensitively? End-of-life assessment, can we use it to intervene quicker? Matt and Ellie both mentioned that these type of setups will generate enormous amounts of data, and so that all of that needs to be considered. Matt did a good job of walking through kind of the infrastructure that you need to think about. But even before the onboarding process and during the onboarding process, it's very important to work through with the data analytics team. Are you using the vendor to do this? Are you using a cloud to do this? Is there internal storage? You need to loop in your IT teams, your analytic teams, and often if there is a huge need to do internal analytics, you're talking about organizational changes where you're likely going to need to embed data scientists throughout your organization, perhaps not only in like a business technology solutions group, but also in therapeutic areas throughout the organization. So that will be very important to help, kind of take apart the data and understand and help develop these studies. It's important in advance to map out the data flow. Again, as Matt touched on, where you're going to store it, how you're going to maintain it. Video data can be very expensive to maintain over time. So do you have enough physical space? Do you have enough budget to store that? Do you have access to the data? Do you have to pay access, from the technology provider to get the data? These are all things that your group should think about. And finally, the real power in this is to be able to take all of this data you're generating from these live animal studies and compare them across the board to omics types of analysis, to pathology, to clinical data. This is the real power of where this is going to go in the future. So enrolling those teams all throughout the organization in a collaborative effort is going to be extraordinarily important. One of the most exciting things opportunities that this technology offers is the ability to translate or even reverse translate from the clinic. Can we help optimize animal modeling? Can we take what is being measured in the clinic and actually be able to use similar types of equipment or similar digital outputs to be able to optimize a model to help us kind of mitigate the attrition that occurs in clinics? So I'll walk you through an example. Itch dermatology and itch model, for example. In the clinics, they're measuring itch for sure, but more so they're looking at sleep disturbance, they're looking at quality of life factors, and so can we take that information in these wearables that people are using in the clinics and somehow retro translate that into mouse or rodent models? Can we look at quality of life factors? Can we look at circadian rhythm disturbances? Can we look at social interaction? all of those things. Can that help make this, more translatable? So enrolling your clinical team early on in the process during onboarding is very important. And, finally, the last slide here, understandably so, certainly onboarding this equipment will definitely impact budget and financial planning. So considerations are, are there purchase options versus lease options? Matt talked about vendor upgrades, PM and upkeep, software updates. Do you need to purchase, that through a subscription? Is that part of your, original contract? Is it going to be an internal development? So all of these things need to be worked out during the onboarding process. And prior to that, you need to assure that you have the appropriate budgets. Again, pilot analysis can take a long time and be expensive. You need to think about the change in workflow practices, what that's going to do to your facility footprints, how that's going to change your support teams. Can as opposed to teams that are traditionally doing phenotyping, can they offset kind of their support to be able to work closely with these new technologies, and then think about equipment depreciation. So that is the last slide.
Awesome. Thank you, Paul, and thank you all speakers. I'm just going to pop in with a few additional slides from my end. So I just want to make sure that if you enjoyed this webinar, if you think that this is useful information and you want to make sure that you get notice of the next webinar straight to your inbox, I do encourage you to subscribe to our newsletter, which comes out twice a month. We have one focused on our resources and one on other resources. Also, if you think, this is great information, you want to help support more of these efforts, you can always encourage your institution to become an individual or an institutional member. You can donate personally, etc. And then I just do want to thank our current institutional sponsors, because they do make efforts like this possible. But now I want to go ahead and invite our presenters to go ahead and turn on their cameras, turn on their microphones, well, at least when they're answering the questions, and then I will get the questions. So yeah, feel free to put them in the Q&A, everybody. So the first question I have is, how do you see the use/challenge of using open source software in the light of the need for reproducibility of data? This question is specifically for Ellie in relationship to Deep Lab Cut.
Well, at least with Calico, we are publishing our code base for any algorithms because we want there to be transparency. And in the past, we've had concerns with what data was being output and how they were analyzing it. And so I'm actually not super duper familiar with analyzing data. I'm not a data scientist, so I can't speak to the data integrity, but I do know that for many, there is a major concern in having direct access to the data, as well as to the video files. And so that's something to consider for your institution if your research team wants to have that access.
Thank you, Ellie. Matt or Paul, anything to add in on that? Okay. Nope. Next question, I think is for Matt, is can the UID system detect individual mice in a group housed together?
Yes, it can. And Paul mentioned this slightly. It's about the density of the cage. There is a sweet spot for every technology, from one to multiple animals housed in the area. But for the UID system, we've done up to 5 animals individually track data, but our sweet spot is around 3 animals per cage based on the density and the data that we collect for it.
Okay, and so then to just follow up the two, so can the antenna read the RFID tag in less than a second as mice move super fast?
Yeah, a great question. Yeah, so it does. We scan, our system scans a cage about every second. So as the mice move within a second, we can probably pick them up.
Awesome. Is there any impact on the RFID chip signal if it is exposed to EMF for a long time?
I could take that one too. Since we use that, the answer for RFID world, the answer is no. With all the technology for the RF side of here, MRI we've tested it, plus MRI 12T, CT X-ray, all fine as well. Ultrasound and even bioluminescence has been fine. So RFID tags are pretty well accepted in that part.
Awesome. If this was to be used throughout a unit, how could it be scaled up with so many cameras, and would it increase the power consumption / carbon footprint of the facility? And so I think really anybody could take this question.
I could probably help. I know Paul had mentioned the cameras shortly. That's more. I'll leave that. But really, these systems don't use a lot of power, which is nice. That's not the biggest footprint. The footprint comes in when we start storing the data. That's where the power comes in. So actually using it, Ellie mentioned in her talk that you got to have power cords for every cage typically, or every rack, I'm sorry. So that does there. But the power usage isn't that great. But really the carbon footprint, I think personally myself, comes into the data storage aspect of it and how you do that, where you do it. Is it better to go to the cloud or do it locally hosted?
Yeah, thank you, Matt. Paul, go ahead.
Yeah, no, I agree completely. It's the power consumption is definitely the data storage, but this is kind of the art in the process of developing out these systems, and where we're at today is probably not where we're going to end up in a few years. So continual refinement. The question is whether you're looking at this point at targeted use of these, or whether you're going to deploy these throughout your entire facility, and that the jury's still out on, but there are some major refinements recently, and it's impressive where these tech providers are coming to, and the ability to kind of pack these systems together.
Awesome. Ellie, anything to add to that?
No, I mean, from an operations standpoint, like using a disposable system, if you're looking at not necessarily carbon footprint, but sustainability, it would be my bigger concern. But I think that for those institutions that are already using disposable caging, then it's sort of like a null impact.
Yeah, and as far as overall, sorry to jump in again. I mean, is there a possibility that we could actually use less animals if we have more sensitive inputs? So, again, these are all things to figure out.
Absolutely. Okay, next question. In your experience, does this technology interfere with the other monitoring systems, for example, Edstrom, Watchdog?
I can jump to that. Most tech providers, they know their technology well, so they design it so it doesn't interfere. There are some very specialized applications that it could. So what my recommendation would be is to talk to the provider. Everything that could be used in that facility, from the software, from the Wi-Fi to any other magnetical fields you have, talk to the provider, and they know their system inside and out, and they know what interferes or what shouldn't interfere with it. So again, that's more of a conversation to have with each provider.
Yeah. Next question is, what's a typical abnormality that the AI will pick up on? Is it simply movement that's monitors, or other factors, i.e. how often the mice interact with each other? Maybe you all can kind of talk from your experience, because I know you have all different systems. Matt, do you want to go first?
Yeah, so it depends. We don't use a lot of AI in our system. That's more in the video-based systems, and really it's how well designed those are. And there's a lot of work being done, open source code right now on the AI. And it's a very exciting field, and there's a whole new field that's being generated by the technology that's being created. So it's really fun to do, but it can be very precise. It take a lot of time. There's learning algorithms. A lot of companies now can learn behaviors, and it can teach them behaviors. So one of the parts was storing all the data. So there are systems out there now that when it sees behavior, it'll start recording and not record the rest of the time. So it helps limit that amount of data that can be stored. So again, those are great advances that as providers we hear from people like Ellie and Paul, hey, storage is an issue. How do we do it? So that's happening. And luckily, a lot of it's open source now too. So it helps everybody out.
Yeah. Ellie, do you want to add on to that?
No, I don't think I have anything else to add. Yeah.
Okay. Okay. Paul?
Yeah, I mean, I think this is, this is one of the most exciting opportunities, in my mind is, can we think about building a toolkit of digital biomarkers, right? And can we develop a constellation of them to apply to different models depending on what you're trying to study? So, AI can be useful in video applications and in other applications, to help us look at micro movements, not only just gross activity, but things like grimace, posturing, gait, social interaction, thigmotactic behavior. There's a whole big list of things that are possible out there, and as this technology continues to refine, that toolkit will become larger and larger. So it's pretty exciting. It depends on the use case scenario and the technology you're using.
Great. Next question. All speakers talked about the challenge getting different parts of an organization on board with adopting this technology. How would they rate overall resistance and difficulties versus the benefits at this moment? And I'm sure that's a it depends answer, but who wants to go first? Ellie?
I can start. Yeah, I can start. I wouldn't say that there's resistance. It's that a lot of folks are not aware of the value. And so for those of us who are championing technology and refinement, we really have to both create the business case and the scientific case. As much as a veterinarian, I really push for animal welfare. If I don't have the business case and I don't have the scientific to back me up, then it's not going to move forward. And one of the things that I've been lucky to have is a group of scientists here at Calico who really champion that cause. And so it really is a group effort to push the technology forward. And so really identifying champions and making that case is probably the biggest challenge to keep the momentum going and to keep people engaged, because it's easy to pitch something once, but it's more of a marathon. Like we've all been in this field a long time and have seen technologies mature, but it's going to take persistent effort, continued piloting, more exploration, and we have to treat it like tech. Not everything we throw at the wall is going to stick, but we have to keep going.
Yeah. Anything to add, Matt or Paul?
I think from a tech provider's perspective, a lot of it's putting the cart before the horse. The labs, the facilities, they want to test it and they want to try it to make sure that it validates the science. They want to work with it before they actually buy it. So that's a kind of an issue you got to overcome. But most providers now that I know of all work with the companies to make sure that's easy to do. Again, it involves IT, it involves piloting the system, how best to do that, and that's where I mentioned that the better you can kind of design your pilot studies around it, answer the most questions you can so that decision is made quickly, helps everybody out, right? Because it's not again, like as Paul mentioned, it's not an easy process. It takes a long time here. But the problems are all solvable. IT solvable, onboarding solvable. It just understand that it's not a plug and play system. That's going to take some work. But once that it is involved, it's amazing what labs start doing with the technology. It blows my mind what I see coming out from just all of the providers. It's amazing. It's an exciting time to be in, I think, to be a tech provider as well as a researcher.
Definitely. Paul, go ahead.
Yeah, I mean, ditto to all the above. The only thing I would add is it's not really a matter of resistance. It's a matter of, this will revolutionize the way we do preclinical research. So it's a matter of timing. When is the appropriate time to thoroughly engage in this? And that's really the discussion that I have with our leadership is not is it necessary, because it eventually will, be definitely useful, but when is the best time to invest?
Yeah, Definitely. And I will say also to that question that in our recent publication, we have a figure, and we go over that figure in the last webinar that provides all kind of like the long list of all of the benefits. And so I'll make sure to send out our publication to all the attendees so that they can see that as well. And you can always use that internally. Joanna asked for links, and I see Harm put a link to the Deep Lab cut in the chat, so thank you, Harm. We'll also be kind of expanding, we're talking about as a group to add more publications and links on our website so that they're easily accessible for that question. Paul and Ellie, do your teams collaborate with colleagues on the clinical side who are developing and using endpoints to share learnings and adopt reverse translation to make sure preclinical biomarkers are clinically relevant? Who wants to go first? Paul, you can go ahead.
I can go first. Yeah, I mean, yeah, no, I appreciate the question. And, I think it's early in the process, and it is certainly our desire in every instance to do that. Ultimately, that's the kind of the meat and potatoes of why we're doing this. So to be able to engage with those groups directly is an important, process in developing out models. I can't be completely honest and say, yes, we do that in every instance, but sometimes there's indirect communication. But the goal is to plug those groups in directly from the very start, and get that process rolling.
Ellie?
Yeah, so we have a pharmacology team here, and others that do sort of learn about what we are developing in-house as well as looking at externally. I could say for Calico, we study aging, and so there is already clinical biomarkers of frailty, which we're also looking at. However, things like frailty are very objective, and there's many different measurements, and Paul spoke to building a constellation of phenotypes to see what is actually clinically translatable. We still don't know. And so while I would like to say, for studying neurological disease, these are these biomarkers of cognition, it's really hard to say, like, well, what's the perfect cognitive phenotype to look at a mouse? And so from a baseline, I like looking at quality of life. I'm very interested in nesting material as a biomarker of health. That there's lots of information on that, but not a lot of machine learning applied to it. And so really teaming with sort of the veterinary side, I think it makes sense to make those connections within the companies. Yeah.
Great. And then the question is, how much training of staff is needed? I'm not sure if this is directed for a particular staff or just kind of, I don't know if you can kind of speak to that. Anybody can. Ellie, go ahead.
Yeah, I can. It depends. As an example, my 9-year-old can operate the iPad like way back, like he knows all the shortcuts and whatnot, and can navigate websites in a way that like blows my mind. And so I think it really depends on somebody's personal use and comfort level with technology. And so I've seen folks, and this is not an age or a generation thing. It's like people who are really adept at technology adopt it pretty quickly and are really good at just sort of feeling out the software. But folks who are less adept take more training. So yeah, but I mean, at the technician level, absolutely. Folks are very trainable, very excited about using this technology and see the benefit in their day-to-day.
Yeah, definitely. Anything to add, Paul or Matt? Again, the question was about husbandry and researchers, but I think the same applies.
I agree with Ellie. Yeah, it's across the board. Learning the systems is not that difficult if you're tech savvy. There is computer systems you have to learn, the new softwares you have to learn. The husbandry side is standard husbandry practices that we see here. So the training it's not as the training. I don't think the training is a hurdle that has stopped anybody. It's just part of the process of getting on board any new technology we're looking at.
Yeah, I have nothing else to add. I think I agree.
Yeah.
Oh, one more thing I did want to add was that, if you're having difficulty with your software, your user interface, reach out to your technology provider, because they want you to be able to scale these systems, and you're not going to be able to scale these systems if it's not something that's user friendly. And so they need to hear that feedback, and most likely they're very willing to oblige with putting it in their queue of improvements for their next release.
Yeah. Next question, potentially a good one, is, at the moment, in the opinion of the experts, is the video-based technology favored / considered superior over the RFID-based technology? And maybe I'll kind of rephrase that to just, if you're weighing video versus RFID, where do certain systems have advantages over the others? Maybe Matt, you can start.
I can start. Yeah, it depends. They both do their job very well. When I mentioned, apples to apples, you can't compare a video tracking system to RFID. It's just, you can't compare them. It's not. And so, but with RFID, you can get grouped house animals easier and get other data such as potential, like a temperature or location of the animal. So it depends what you're looking for. What is your endpoint? What do you want? What's important to your research? Those are the questions you have to kind of figure out. All the technology, they're great technologies, and one isn't superior to the other. It's just, they're just different, right? They're all going to be different. So look at what you're looking for and how granular do you want to get with your data set, and then try the different technologies, see what is going to fit best with that. And again, most providers that are on this system, they're happy to have trials head to head, because we all know there isn't one perfect system, right? So is what fits best to your research.
Yeah, and I'll jump in and say, in our manuscript, we have a downloadable table, again, where you can kind of write out all the things that you're looking for from a technology, and then think about, then compare to the individual technology. And in the technology hub that we're going to be creating, hopefully by the end of the year, we're going to have some filters such as housing density, such as the particular biomarkers that you're trying to collect, etc. Ellie or Paul, do you want to chime in on that question? Ellie, you can go first.
Yeah, sure. It really depends on what the scientists are looking at, but I will say that we have multiple modalities of smart caging, because if it's a metabolic readout, we're going to need those metabolic cages. If we're looking at temperature, you really could only do that with RFID. And so they all have their very specific use case. I also think there's power in looking in different angles. The only limitation for video for me is the single housing, but with that paper that I showed with group housing and tracking, I think that is coming. We just need to keep working towards that future.
Yeah, definitely. Paul, anything to add?
Yeah, I mean, it's unlikely at this state of technology that it's going to be a one size fits all. And an organization may want to consider a hybrid approach where, some applications with RFID are very important. Some with video are very important. So, you have to look at that, what your needs are, what your case use is, and plan from there.
Yeah, definitely. Any other questions from participants? Yeah, and then we have an additional comment. Some people are putting some podcasts. Cindy Buckmaster's Get Real podcast has some good stuff. We actually do a podcast as well called the 3-Minute 3 Rs that highlights 3 publications in a minute each. Another speaker or another individual of our initiative chimed in on the discussion. RFID can be powerful for animal identification in group housing. Video can be very powerful for movement tracking and behavior. Combination can be the best of both worlds. And there are tech providers that are kind of working on that integration. And then again, it really, like all the speaker says, it depends. It depends on your what you're looking for. So definitely, you have to make those hard decisions and have those hard thoughts first. Any other questions from participants?
I want to expand on
go ahead.
comment. Perfect comment, Lucas. Thank you for that. I mentioned earlier, we don't know where we're going to be in 3 years or what we're researching. and the technology is the same way, where it's being developed, you know, every month, every 6 months. You know, we're working with all the vendors are working together. It's amazing, a collaborative group effort that the vendors are working together, the tech providers are working together. So that's exciting for us as a side, knowing what we're creating, and in the next year or 2 years or 3 years, what the providers can push out to the scientists, let them start testing. So that's why I was saying, you know, look for the adaptable system. Always ask the questions to the tech providers. Push them. That's why I say push it. That's how we grow, as you know. And Paul is as electrical engineering side to get in there. You like getting those questions and challenging those mind and seeing where you can push technology, and that just makes it better for everybody. So it's exciting times.
Yeah, definitely. Another participant asks what is concerned with invasiveness of RFID tags, and asks if you always have to use RFID tags with translational digital biotechnologies.
They're very non-invasive. A lot of the providers, the Jackson Labs, Charles Rivers, the Innoatives of the world can put them in for you. So that's a pretty straight. It's a very non-invasive. Typically, I don't know if any because Ellie and Paul, you may answer this more, but I don't think it's considered an invasive surgery at all. A microchip implant, I've never seen it. It's very sub-Q, simple implant. So to use the RFID tag, you would use the tag. There have been like collars people put on rodents, large. Sometimes you can do it in large animals too. If you want to use the technology in dogs or even larger in primates as well, it could happen. So it's a very non-invasive process for RFID and simple. And it's been around for a long time.
I would also like to add that even if you're not using the UID system, for example, if you're using video or any other caging, it's very critical to have ID on the mouse to make sure that you are recording from the correct subject, the correct age, the correct group. In our experience, at least, the RFIDs, we have less loss, whereas ear tags, we might lose them. And so for longitudinal studies, you have to consider which ID method will ensure that you're recording from the correct animal, and you won't have to replace. So you have to offset that. I could either ear tag twice, or I could just put an RFID in. And so it's that cost benefit that you'll have to do depending on the study that you're running.
Yeah. Paul, anything to add? Yeah, and just to, from our experience, the RFIDs are tolerated very well by the mice. They don't seem to mind them. And yeah, what Ellie was saying, kind of not, you know, if we're thinking large scale, thinking about the 3 Rs, losing data from a longitudinal study, but from a resource intensive study can be really, really negative from that 3 Rs perspective in terms of reduction. And so sometimes having these good identification methods can be really critical. Any other questions from participants? I'll wait just a couple minutes. Any other thoughts, comments from the presenters as we're reaching the end of our webinar? Okay, well, as I see no more questions, I will go ahead and wrap up today. I want to thank everybody who attended live, as well as those who may be watching the recording. It was great to have you. And again, stay tuned for our next webinar on study design. We're going to be releasing the date and time and registration page of this soon. And we will be releasing the recording of this, posting it on our website for reference and sharing with colleagues. So thank you again. Have a great rest of your day. And yeah, if you want to engage with NA3RC or our initiative, please just reach out to me. I'm happy to talk anytime. And I know all of our speakers are very happy to talk at any time as well. Okay, goodbye, everybody.
Thank you, everybody.